Okay, today we're going to start talking about genome editing with the CRISPR system, and I highly recommend that you watch the video. The URL is given here by Jennifer Doudna that explains the origins of this system. She and Emmanuel Charpentier won the Nobel Prize in Chemistry this year as a result of their developing this amazing technology for genome editing. So what CRISPR is in its natural habitat, which is to say bacteria, they form something of an immune system. The acronym CRISPR stands for Clustered Regularly Spaced Short Palindromic Repeats. So in bacteria, these are DNA memories of previous phage or foreign invaders from some progenitor cell, that they're little snips of DNA arranged in a particular order in a row, which enables the cell to recognize when the invader reappears as a way of fighting back and destroying the invading DNA. So the CRISPR is a locus, a genetic locus of DNA transcribed to form a guide that will then recognize complementary DNA and destroy it. This involves an RNA called the tracer RNA and a protein nuclease called the Cas protein. Here's a diagram from their website or her website representing a prokaryotic cell which is about to be infected with a bacteriophage or attempted infection by a bacteriophage. So what's represented here is the CRISPR locus itself, and it consists of some standard important parts. Like first of all, there's what's called the repeat region, shown as these dark triangles, diamonds, interspersed with so-called spacer sequences. Now the spacers are actually the business end of this, because that's what's going to guide the CRISPR-Cas9 protein to the foreign DNA in order to selectively cut the DNA and inactivate it. So the CRISPR locus itself consists of repeats of a RNA encoding portion, which is common to all the CRISPR pieces that we will eventually get, punctuated by spacers, which represent the DNA that we'll be able to identify and cleave and protect ourselves from. So CRISPR is transcribed, processed to form these small little things, which consist of the RNA, which is will serve as a guide, but also a standard part of the RNA, which will bind to the Cas9 protein and allows cleavage of the corresponding DNA. So what's represented here with the different colors are different pieces of DNA, different sequences of DNA that are specific to previously known invaders that we can now use to protect ourselves. So what's represented in the upper right is a bacteriophage, which is attempting to infect the cell. And the CRISPR, there's a green CRISPR that matches a green DNA, which is going to then bind to, in a complementary way, the RNA will bind, allowing the Cas9 to cleave the DNA, making a double-strand break, thereby inactivating it. So this is naturally part of the bacterial cells and is a very good way for them to protect themselves from viral invaders. Now, it didn't take anyone too long to figure out that if you had a specific sequence-directed nuclease like this, that we could use this for our very own purposes. And our simple view would be that we would have, of course, the Cas9 protein itself, which we can produce easily, and then the guide RNA arranged as a so-called scaffold, along with our spacer, which we would design in order to target whatever nucleic acid we want to cut using the Cas9 protein. So these protein and RNA components will form a complex, which will then be able to hunt down, identify, and cut any corresponding complementary DNA sequence. Okay, so here's a very commonly seen representation of this situation. One important sequence to recognize here is called the protospacer adjacent motif, or PAM. The Cas9 protein has not much by way of sequence requirements, but it does require that you have a sequence NGG immediately adjacent to the sequence that you're going to try to cut. Now notice how this is represented, though. You've got your target DNA double-stranded, sort of opened up for the purpose of cutting. The beige glob in the background is the Cas9 protein. But importantly, you've got a guide RNA sequence, which is the one that you would design to target the region that you want to cut. The little scissors indicate where the cut sites are going to be. And of course, each one is going to have the tracer RNA, which will be built into your vector, basically, so that when you clone in the DNA that corresponds to your intended guide, you will eventually produce this guide RNA that's specific towards whatever it is you're going to try to cut. Notice that both strands are cut, however. Notice also that your guide is going to anneal to, hybridize to one of the strands, and it does actually itself equal in sequence the upper strand where the sequence isn't shown. And that's going to be important in terms of designing these things later. Okay, so some of the vocabulary: CRISPR, Cas is a Cas9 associated protein. Okay, this is a nuclease. It actually has two nuclease activities associated with it. The one from bacteria, they're all bacterial, but S. pyogenes is the most commonly used one, but there are variants on this now. Okay, importantly, you've got your SG or guide RNA, and that's a fusion between, these were actually artificially put together to form the tracer RNA, but it consists of a fusion between the bacterial RNAs that are part of the CRISPR system itself and our chosen spacer. Our spacer is going to choose the DNA that's going to be cut. So this is going to involve a spacer of about 20 nucleotides long, which is sufficient to bind firmly and specifically, usually, to the region you're trying to target. Okay, so the PAM or protospacer adjacent motif is important because the Cas9 needs this sequence just 3' to the sequence that's going to be the same as the guide. Now, it's not going to be present in the spacer that you're putting, but it will be there in the genomic DNA, and it's an important part of the nuclease process. Here's our picture again, showing the targeted locus. Both strands will cleave, the location of the PAM, the location of the guide, and what's going to be cleaved. So what happens in a cell, this is, of course, going to create a double-strand break because you've cut both strands of the target DNA. The cell will naturally race to try to fix that. A double-strand break is lethal to cells, and there's two common pathways that are going to be under consideration here. The first and easiest is the non-homologous end-joining pathway, which results in deletion and occasionally insertion of a few nucleotides at the side of the break. So this will tend to knock out the gene if it occurs in an essential portion of the gene. So the first line of defense the cell has is this non-homologous end-joining pathway, which basically acts as double-stranded breaks in order to put the two pieces back together again. The other one that's going to be relevant later is a homology-dependent repair. Okay, so this will occur, and this is the most powerful part of this, that if a cell contains the donor DNA that can be used as a template to repair by resynthesizing the region. So it's through the use of these homology-dependent repair oligonucleotides or PCR products that one is able to actually replace the sequence that's present in the chromosome with whatever else we'd rather have there. So this is a very, very huge and important process because you can do things like tag proteins in the cell. You can fix mutations potentially. Very many different things you can do when you've got the choice of what oligo you're going to use to repair whatever break that you specifically made. Examples later. Okay, so double-strand breaks are dangerous, to say lethal, and we have a lot of proteins that can fix them. Okay, and I'm going to describe that pathway very shortly. And then, of course, then we have the homology-dependent pathway, which will show in very, very, very overly simplified terms just to show what's going on. Okay, so this is going to be non-homologous end joining, and we are showing this as a lightning bolt, a red lightning bolt, chopping the DNA at a particular location. As a result of that, the ends are frayed and you have a double-strand break. Well, we've got a whole host of proteins that race to the scene to try to fix this, including polymerases, nucleases, ligase, etc. But the upshot is that you generally lose a few nucleotides at the side of the break in the process of trying to put those broken ends back together. Now, you're going to get a variety of different types of deletions, different lengths of deletions often. But in any case, you're going to be usually losing a few nucleotides, which can have the effect of inactivating that gene. So these ends need to be dealt with, and the cell has very powerful ways of doing that because double-strand break repair is extremely important. But the process is mutagenic. Okay, if you're going to make mutations, you're going to be deleting a few nucleotides. So that is in fact a deletion or a mutation really in the gene, which could be important if the deletion is within an exon. So this is usually depicted simply double-strand break on the left-hand part of the slide. One pathway for its repair is a non-homologous end joining pathway, with the result that the DNA is fixed. It's not a lethal event anymore, but you will end up with deletion of a few nucleotides, sometimes more than that, with the idea that many times your gene will be disrupted. The homology-dependent repair is a different process. So here's a very simple-minded view of the double-strand break repair pathway, that you have the break, you have processing by the cellular enzymes, and then you have end joining by DNA ligation. But so the repair has been made, but you lose nucleotides. Okay, so the other pathway is homology-directed DNA repair. Okay, so this is the way we can add sequences or fix mutations that we don't like. So the idea is if you have a break, the cell has the option of processing that break by getting a template DNA that it can copy to replace not just the break itself, but the region surrounding it. So if we have a DNA oligo or PCR product that is homologous to the break on both sides, the cell can use that to replace whatever region is broken. And this can be actually a fairly extensive piece and can lead to gene replacement, in which we can fix the double-stranded break along with fixing any mutations that we don't like that are at that particular location, among other possibilities. Okay, so here's our homology-dependent repair pathway, which is not shown very well at all, where we have the break, and then we are synthesizing, we're using that homologous piece of DNA as a way of replacing what's there, and we can replace it not just with what used to be there, but what we want to put there in the form of epitope tags and various other sorts of things. Okay, so we can exploit this to hunt down specific complementary sequences, make double-strand cuts, and then for whatever purpose, and I'm going to give a few examples. Can be done in cell lines, but it also can be done in whole organisms. Okay, but first of all, once you've got your guide RNA designed and targeted, you have to get it into a cell in order for the CRISPR system to do its work. Okay, so this is kind of familiar territory to us right now because you can deliver the CRISPR in the form of DNA, RNA, proteins, etc., by all the usual players such as electroporation, transfection, injection. People can deliver it by means of plasmids or by means of viruses. So it's like any other delivery of nucleic acids to cells that we've seen before in terms of transfection. Think about short hairpin RNAs, silencing RNAs, etc. Okay, so we can represent that as a picture here, like we can deliver CRISPR components by means of a plasmid. In one set of ways of doing this, one can purchase plasmids which actually encode the Cas9 protein, which has been engineered in such that it contains a nuclear localization signal for mammals, for example, and within the same molecule encode the intended guide RNA adjacent to the structural components of the CRISPR complex. So the DNA-based delivery is a little more complicated from the standpoint of the cell because the plasmid has got to get into the nucleus, has to get transcribed. The RNAs have to get out to the cytosol for translation into protein, but they do. But the cell will actually form the correct Cas9 complex, which can then go back into the nucleus to adjust, to cut its genomic target. Alternatively, some people will make the Cas9 guide RNA complex from purified proteins and RNA in vitro and then deliver that directly to cells. So there are many options as far as how this sort of system can get delivered into whatever cells. Okay, same idea. Electroporation, we haven't talked about that much in the past, involves basically an electric shock to the cells that causes transient membrane permeability so that nucleic acids and proteins can then get in. Or else you can deliver the materials by means of a lipid nanoparticle of whatever sort or another. So the gene delivery is very much like whatever we've seen before for delivery of nucleic acids generally. Okay, so we can have electroporation, we can have transfection with a transfection reagent, lipid nanoparticles, and we haven't talked much about viral vectors and ligand-mediated interactions, but those can be used too. But this is kind of standard as far as getting things into cells. Easier for mammalian than it is for bacteria and fungi. So some people will purchase the protein, the Cas9, but many labs will actually purify their own. The genes available, expression plasmids are available with tags, so it's not hard to purify that protein at all from E. coli. Okay, so you need to, of course, design your guide RNA specific to your experiment, but those can be made in vitro. They can be commercially made for you, or else whatever. But you can put them together, and they will associate to form a complex, which can be transfected by normal methods. So none of this is difficult. Okay, so the plasmid deliveries, of course, you're going to be cloning in E. coli. So of course you have to have origin selection for E. coli, and you need to have restriction sites to open your vector to get your target sequence cloned, etc. But these vectors are made so that they already will encode the Cas9 protein, and basically kind of it's a one plasmid thing that does everything for you once it gets into the cell. Okay, so here's an example of one of those, the all-in-one vector. It's got a region that encodes the Cas9. It's got a promoter that works in mammalian cells. It's got a nuclear localization signal added to the protein. And of course, you've got a region where you can clone what you want for your guide RNA, and a region that encodes the guide RNA scaffold. So these are fairly easy to use and easy to acquire. Okay, so what do we want to edit? Well, there's a lot of different things we want to edit. But the recent famous example is the beginnings of what we hope is an extensive improvement in the possibility of human gene therapy. So this example is going to be to treat sickle cell disease, and in some sense is a cure. Now this is a somatic thing, it's not a germline thing in people, but it appears to be very successful. And people have tried different approaches. Different approaches are currently underway to get this to work. And so far, it has been amazingly successful so far, at least some of the approaches. Okay, so the ones that have come along the furthest are ones that use an interesting phenotype or interesting genetic syndrome called hereditary persistence of fetal hemoglobin in order to treat and possibly cure the sickle cell disease. Okay, so what this is, and it's an unusual situation where it's been noticed for a long time that people that had sickle cell disease had a much, much, much milder form if they also had this other hereditary persistence of fetal hemoglobin mutation. And there are many of these. Okay, this does sound strange, but it does work. Okay, so we know that in mammals, different hemoglobin beta chains are made at different times during embryonic, fetal, and adult life. So our globin gene organization is as like that. We have the globin genes arrayed in order in a particular location, and they are activated in order during development. Now, normally, shortly after birth or around birth, there is a switch from the fetal hemoglobin form to the adult forms, and those are the ones that are going to persist throughout life. But in cases where there are mutations leading to hereditary persistence of fetal hemoglobin, the switch doesn't occur, so that the fetal hemoglobins are not turned off, and they will persist throughout life. So this is a trick because it apparently has been known to mitigate the effects of a sickle cell allele. So this trick is what's going to be used in these gene therapy experiments to try to mitigate or ameliorate the symptoms of sickle cell disease. Okay, so here's what fetal hemoglobin looks like in people, right? We have a very early embryonic form, which is not shown in this diagram, but hemoglobin, the fetal forms, persist throughout development until around the time of birth. At this point, the adult hemoglobins are switched on, and it's after that point, when the fetal hemoglobins have been turned off, that sickle cell symptoms can start to appear. Okay, so the people that have hereditary persistence of fetal hemoglobin, they are just fine. It's a syndrome, but it doesn't cause any major problems. And so what people decided to try to do was to create the persistent phenotype in people with sickle cell disease, hoping that it would mitigate the effects of their disease. So we're essentially creating a genetic syndrome in cells and then in people cells in order to mitigate these effects. Now, reasons to think that this is likely to work. This represents, I'm not sure how they're measuring morbidities, but it relates how morbidities of some sort or ill effects, whatever, related to the amount of fetal hemoglobin one has. Now you can see there's a negative slope to this line, and what it's telling us is that the more fetal hemoglobin is produced, the less the symptoms are serious. So somebody that has no fetal hemoglobin at all will have very severe symptoms, and the symptoms get less and less and less according to how much actual fetal hemoglobin is being produced, telling us that the fetal hemoglobin can make life a lot easier for people with sickle cell disease. So mutations, what has been used and how to create them. There's a couple of different approaches, but they all focus on this particular repressor protein called BCL11A. So this repressor protein, BCL11A, is responsible for this switch. It binds to the promoter enhancer region adjacent to fetal hemoglobins, two of them, A gamma and G gamma, shutting them off. So if you want to fix or create the persistence phenotype, you've got two basic ways to do that. One is that you can mutate the BCL11 binding site near the A gamma and G gamma regions, and so that repression won't occur. So if the BCL11 protein can't bind, repression will not occur, and the fetal form will persist. The other approach, basically the same thing, is to use CRISPR to mutate the erythroid-specific region of the promoter enhancer of BCL11 itself so that it's not able to be active in blood cells. So you can either mutate the site or mutate the gene encoding the protein, but the effect is supposed to be the same, that without that repressor, you will not shut off the fetal hemoglobins, and they will persist, and this will mitigate the effects of the sickle cell disease. So here's a picture of the fetal hemoglobin region, and it describes some essential features of this. One is, this is the same region exists in front of both the A gamma and the G gamma, because we've got two fetal hemoglobins, one of each. There's a region called the delta 13, which is a deletion, a naturally occurring deletion, which produces hereditary persistence of fetal hemoglobin. So that's the one important target region for this, because we know that that one small deletion is enough to cause a persistence phenotype. There's also other mutations in the population which lead to point mutations, and importantly in red is the BCL11A binding site. So there's a lot of concordance of information here. The binding site is actually overlapping with the naturally occurring deletion, indicating that this part of the gene is what we want to look at if we want to create the persistence phenotype in cell lines or in people's cells. So those are the target regions that we're going to be concerned with. All right. So I think I just said this. The binding site is part of the sequence, and the delta 13 is a very short deletion compared to a lot of others that cause the same syndrome, and that we know that those are good targets for reasonable targets to try and mimic this in cell lines to try to see if we can create this particular persistence phenotype in cells, and then maybe move on to produce the same effect in people. So here's a diagram of the region. You've got the HBG2 and B1, G1 are what we have been calling the A gamma and G gamma. Those are the fetal hemoglobins. The ones on the right are the adult hemoglobins. On the left, of course, is the embryonic one. The part of the sequence here that is in the box, that's the 13 nucleotide deletion. And the BCL11A binding site is adjacent on the left end, but adjacent to this sequence and overlapping with it. So that's the reason we're going to want to target with our guide RNAs to try to cut it and then destroy that binding site in order to mimic the persistence phenotype that we know we would get with this 13 nucleotide deletion. Okay, so the guide RNAs that were used, this is 2 of them from 2 different papers. Okay, what those do is they target that region in order to create the double-strand break that's going to cause deletion of a few nucleotides that will hopefully cause a persistence phenotype that we can then pursue further. So this was done in a cell line. The CRISPRs were engineered and introduced into a cell line, and then analyzed for whether they caused expression of fetal hemoglobin and for whatever deletions that they might have. Now, all of the constructs or all these sequences come from separate transformants or transfectants, and they all had at least some deletions, as indicated by the dashes, indicate those nucleotides are missing. And they all overlap with the region that we know, the 13 nucleotide deletion in real life, and with the BCL11 binding site. So these do cause significant expression of fetal hemoglobin, and they do have small deletions as we were expecting. And one of them actually does exactly mimic the 13 nucleotide deletion that's been found in human populations. Okay, so further evidence that this is going to work is using cells from a sickle cell patient that were produced. They were isolated and then treated with the Cas9 protein plus the guide RNA. And the cells were exposed to hypoxia, which is a situation that causes sickling to occur. So cells that were transfected just with the Cas9 protein itself sickled with very high frequency, as indicated by on the left, in the cells that are just expressing Cas9 but without the guide RNA. Right? Another population of cells got the guide RNA along with Cas9, and the sickling was greatly reduced to less than 5%. So at least in cell lines, or in cells from people, cell lines from people, this does work. It does not just allow the production of fetal hemoglobin, but it also reduces the sickling phenotype in their cells. So this is further evidence that this is going to be successful. Okay, so they're measuring this in cell lines, measuring the percent of the hemoglobin that is actually fetal hemoglobin. And cells with this Cas9 only have a very small amount, as you would expect, which is greatly elevated in cells that are carrying the guide RNA number 1 plus the Cas9 protein. So fetal hemoglobin is expressed in these cells in response to the presence of this guide. So these cells are mimicking the hereditary persistence of fetal hemoglobin in these cells. Okay, so other approaches which haven't quite gotten so far is just to fix the mutation using a homology-dependent repair template. That's a good idea too. So that one's still in progress. Okay, so trials in progress so far are ex vivo. So being convinced with these and a bazillion other experiments that this is likely to work, it's being tried in clinical trials with people. So with ex vivo experiments, what we're going to do is take the somatic cells from a person, get the cells out of the person. We're not injecting it directly into the person. We're getting the cells out of the person, treat the cells, and then return them to the person for the purpose of letting them engraft and then do what they're going to do, in this case, to produce fetal hemoglobin. Okay, so this is represented as a strategy here. Okay, what you do is you have cells from the people, and what you're going to try to do is isolate hematopoietic stem cells from the blood of the people. So the first thing is to acquire cells from the person and then manipulate them in vitro using the CRISPR system to try to basically create more fetal hemoglobin in those cells by knocking out the binding site for the BCL11 repressor. So that's what we're doing. And we're then harvesting the cells, characterizing them really well before infusing them back into people. So this is a very hard process for people to go with. It's essentially a bone marrow transfer, and it's extremely hard on the people. But it's been worth it for the people that have had it because it seems to have been successful. So when the cells are placed back in the body, they will engraft, and you will reconstitute the immune system, and then they will follow the people over time to see what hemoglobins they're making proportionally and how much, and also how they're feeling. Okay, so the results, and this is early, there have been more successful experiments since then. What they're doing basically is to start with the person baseline, 74.1% before the infusion of the manipulated cells was the S hemoglobin, which is a sickle cell form. And over time, what's happening is we're looking for the accumulation and maybe even taking over of the hemoglobin F, which is the fetal form. So early on, there's very little of the fetal form. I don't understand this 1 month. It just seems to have hemoglobin A, which is impossible. But then you can see over the months, like by the second month, 25% of the person's hemoglobin is the fetal form. And then that increases over time till approximately 50% or half of the person's hemoglobin is the fetal form. So this does work. And people have been just rejoicing on how miraculous of a life changer this has been for them. The few people that have been treated in this way have been stably much better, no longer require transfusions, no more trips to the emergency room, etc. So this does look like a potential or real cure for this disease. Right, so the one lady that was the first one had been getting 5 blood transfusions a year, crises, many trips to the ER, and after 9 months she's had no crises, no blood transfusions, and she's feeling just fine. So this is very encouraging for everyone, really.